Namaskar, dear friends, colleagues and students. Myself, Professor Naresh Kumar Singh, heading the Department of Veterinary Surgery and Radiology, Faculty of Veterinary and Animal Sciences from Banaras Hindu University. Today, we will discuss a topic that is radiation sensitivity of different body cells and radiation protection. Before we go in details, we would discuss radiation absorption. If we will talk about absorption of energy from radiation in the biological material, it happens in two ways. Any kind of radiation that is being absorbed by biological material, it leads to two different phenomena. The number one, excitation. How does it happen? It happens by raising of an electron in an atom to a higher level. And the another phenomena is the ionization. It is the ejection or ejecting of one or more electron from the atom. The radiation, this kind of radiation is known as ionizing radiation. So during excitation, what happens? Either the number of electrons from an orbit is increased, or the ionization, or it decreases during an ionization, or the electron which is in the stable form. As we know that any atom is having different shells like K, L, M, N and so on, and every orbit has a certain number of electrons. So excitation happens if an atom gains an electrons, whereas the ionization happens when it loses an electron. And ionization is very, very important for a beam or the particles to move in a particular direction. That happens in the X-ray or the radiation. And different types of ionization we will discuss is the direct ionization or the indirect ionization. So whenever the ionization or the beam gets the movement or a momentum, there are two basic forms that would arise when they interact with the cells or the tissues. In the direct ionization, when the absorbed X-ray, they directly cause ionization leading to the damage. Example, the electrons, alpha particles or the beta particles. So they hit the nucleus or the DNA material of the cell, leading to the direct damage, and that gets evinced onto the biological system. The another form is the indirect ionization. When these incident rays are absorbed in material, they give up their energy to produce the fast moving charged particles, and when the fast moving charged particles are produced, they just move around within the cell and damage it, and these are electromagnetic radiations. So it would be better to understand what is an electromagnetic radiation. If you look at this picture, you can see the different number of radiations, right from the radio waves, microwaves, infrared rays, visible light, ultraviolet, X-rays and gamma rays. This is the electromagnetic radiation, where you can see the electric and the magnetic field are perpendicular to each other. And look at the example of the X-rays and the gamma rays. They occupy the short wavelength end of the electromagnetic radiations, and from there we can understand the nature of X-rays and the gamma rays. So the actions of radiation, so as we said, the direct ionization or indirect ionization, in this, when these X-rays actually hits the tissues in the process of making an exposure, during an absorption, what happens? The radiation directly interacts with the target cells, and the atom gets ionized or excited, as we discussed just earlier. Indirect one is when the absorption happens, the radiation interacts with the atoms or the molecules in the cell, particularly the water to produce the free radicals. So how this happens, I'll just show you by way of this picture. Look at this picture. This is the helical picture of the DNA molecule, or where you can see the incident rays which is just falling onto the cells and how they are actually interacting. Look at the lower side. If the incident photons are just falling and they are ejecting one electron out, causing an ionization, and it is just hitting a particular area of the DNA molecule area and making certain changes. So these changes by way of the single chain damage, or single strand damage, or a double strand damage that would happen here, and that gets reflected onto the biological system immediately, on the immediate basis. The another type of interaction that happens, and that brings about the indirect effect onto the cell, is through this way. This is the second picture that you can see here. It is the photons when it is falling and doing an ionization. It is actually affecting a cellular system or the cell, wherein within the cell you have a water, and this water molecules is broken down. So while breaking down, what happens, there is a formation of hydroxyl radicals, or there will be superoxides, and there is a hydroxyl ions. So these are ions, and these are basically the radicals which actually brings about radiolysis. We call it as a radiolysis, or this radiolysis brings about the damage to the cell, causes the cell damage. Okay. Now, when the cell damage, we say it is nothing but the cell death. How the cell death actually happens? There are different forms of cell death that is witnessed as a result of irradiation. It is the number one, we could say it is mitotic death. Then the next one is the interphase death, apoptotic death, or necrotic death, and autophagy. These are 5 different forms of cell death that one could witness as a result of irradiation. In the mitotic death, what happens? The cells lethally injured by the clinically relevant dose of radiation execute one or more divisions before mitotic death. And a mechanism, how does it takes place, is there will be failure of spindle formation in the metaphase. There will be loss of G2 cell cycle or the checkpoints, improper chromosomal segregation due to the damage or the loss of genetic material. So these are the things that would be seen during a mitotic mitosis of a cell, interruption of the cell cycle. The another one is the interphase or interphase death. It is seen in the radiosensitive cells within 2 to 6 hours. And when you can see this picture here, then you have a nucleolus or the nuclear envelope and the chromatin material. So during this, what happens is nucleolus and the nuclear envelope, which is distinct, and chromosomes are thread-like forms. So when this kind of death of a cell happens, it happens within 2 to 6 hours. And if there is a damage during this phase, these kind of cells will never do participation subsequently in mitotic activities. And the death of the cell during this phase happens due to the rapid apoptosis process. The apoptotic death is another form of cell death. In apoptosis, it is a programmed cell death basically. The normal cell that you see here will, upon irradiation, there will be cell shrinkage. The size of the cell will become smaller, and the small size shrinkage will happen basically because of the chromatin condensation, and we call it as a membrane blebbing. It means the actual size, it will just form a shrinkage will go even more further, and the blebbing will appear. This is the blebbing of the cells, which is quite witnessed here. And upon blebbing, the nuclear collapse will happen, and there will be further advanced form of blebbing of the material will be seen. And after the continuous blebbing that is witnessed here, the subsequent stage, what will happen? There will be disintegration. There will be apoptotic body formation will be there, and the parts will get disintegrated, and finally there will be lysis that you can see here. So this is how the apoptotic cell death will happen, and this kind of cell death will never take do the participation in once the cell is totally dead, there will be no chance that it could ever take do the participation in the mitotic activities. The necrotic death, here you can see the cell, the normal cell, in two forms. One, it is a possibility that there is a recovery of these cells could be possible, that this is a reversible injury here. The cell, apart from the chromatin shrinkage, there is a progressive injury which could also be seen, and lot of lysis would happen into the cytoplasm of the cell. This lysis could cause the inflammation of many of the mitochondrial cells, and these are the energy bags or energy sources basically. And if there will be depletion of these things, the powerhouse of the cells, the necrosis will be quite prominent. The last form of cell death is autophagy. Here the cell internalize the cell organelle within the vacuole and digest them. It means it is a self-killing form of cell death. It's a kind of also the similar as apoptotic cell death, which is a programmed cell death, and this is also a programmed cell death, but it is just an opposite of the apoptosis, where in apoptosis the cells die because of the external irradiations, but the autophagy is the one. This also appears, this is an indirect form. The irradiation could bring about certain changes in the cells that they enter into the phase of the autophagy. So these are the different forms that will happen. Here the cells will basically do the internalization or its own organelles, and finally the autophagy will take place, and there will be death of the cell. Radiation injuries are of different types basically. The radiation, or during irradiation, a lot of different tissues gets exposed, and in a different tissue at different point of time, the injuries are being witnessed, and we categorize for our ease into three different forms. Number one, it is the acute response, another is subacute response, and the late response. In the acute response, as we see, the name acute itself, it means it is seen within the 6 to 8 weeks time. And what happens during the acute response is there is a sudden decrease in the stem and the progenitor pools, and where these pools are basically present, irradiation happens. The major pool is in the progenitor cells are in the reproductive organs. The major pool is in the bone marrow regions. So these are the major pools where the stem cells, because of the irradiation, definitely get reduced, and depending upon the severity of injury, that will decide how much of the cell will decrease, and whether the cells which are available will actually enter into a new cycle or a cell cycle. severity will also be directly proportional to the how the response of regeneration would take place in the next cell cycle. So that's all dependent on how much of the severity has happened, and that severity is the reflection of whether it is a mild irradiation or a moderate or a very high dose of radiation is being exposed, to which the patient has been exposed. The another form of response of irradiation that we witness is subacute response. This is seen from a few to several months after irradiation. The symptoms that we come across in this kind of category are reversible form, but sometimes in certain situation where it cannot be controlled and if it has not been taken care of very well, death may happen. And mostly this form of response are seen in the remodeling form. Remodeling means when the cells are in the in a form or a phase where it enters once again into the regeneration form. So no response means or sometimes the somnolence of the brain upon irradiation. So it means it is totally dependent upon if the damage has to happen, the brain is either sleeping or it is not responding back upon irradiation. It means the signals which are going down the line to the various body parts near or far will get affected. At the same time, this kind of subacute response are also seen as a result of irradiation as a subacute pneumonitis after a lung irradiation. Now, in the late response, what happens? It is very slow, basically, seen mainly due to the depletion of proliferating cell. For example, if the exposure has happened in the urinary system where the kidney gets exposed, it has a renal tubule, or its renal tubular epithelium gets affected or irradiated. As a result, what will happen? There will be desquamation of these epithelium, which has important role in the filtration, which hampers the filtration process or the urinary filtration. Oligodendrites, the brain cells, Schwann cells of the nerves, and endothelium and fibroblast are the one which also gets affected upon the moderate to very high dose of irradiation. And however, the response that we get in the patient is quite late, and by the time the response are evinced, the treatment becomes very challenging. As we said, the irradiation when happens, the direct and the indirect ionization or the effect that happens is basically been the principal target is the DNA of the biological system. So you can just see there are different strands in it. The strands, there will be either a double strand break, DBS, or single strand break. So these are two different forms which is normally being seen as a result of irradiation. The single strand break gets repaired, or there is a chance to the sometimes get misrepaired to bring about the mutation, whereas there will be definitely the chromosomal aberrations leading to mutation. Lethal aberrations are the one which has also been aberrations, ring chromosome aberrations, or anaphase bridge, or that is a chromatic aberration. Also being seen, there are we witness two different forms of the symmetrical translocation, wherein break in two different with a broken ends being gets exchanged. That you can see if it is the this is what happens in the symmetrical translocation, whereas the small deletion, the 2 breaks in the formation between the 2 breaks. For example, in this form, there are 2 breaks basically, and chromosome leads to the loss of genetic information between the 2 breaks. So when this there is a immense loss of the genetic information during the process. So these are the depictions. You can see that dicentric in the G chromosomes, there is 1 break in each chromosome, and illegitimate unions and dicentric chromosome plus form. Ring chromosomes, here the pre-replication of the G1 chromosome, there is a break in both the arms of the same chromosome, and there is a which is evidenced in the lethal aberrations of the chromosomes. Anaphase bridge. So in the post-replication unified in a different format as a sister union, whereas the dicentric chromatid plus acentric chromatid is in this form of lethal chromosomal aberration. So classification of radiation damage. So when we see the classification of the radiation damage, the lethal damage and lethal damage is irreversible, irreparable damage that leads to cell death, whereas potential circumstance but can be modified by post-irradiation environmental conditions. Sublethal damage, unless the additional sublethal damage is added. So this is the form of radiation damage, and since also understand that how we can actually reduce the effect of irradiation. So it is in a 2 ways. Basically, the kilovolt potential of the radiation devices or magnetic resonant imaging device or a plain radiograph or an X-ray, which is having a certain capacity from 0 to 25,000 kilovolt. The personals. So the images that you see here is basically the radiation protection devices. So in case of veterinary science, well, the taking an example, the horse or the cows or the buffaloes. So 700 kgs body weight, and they're quite bulky animals, and we cannot sometimes we take it as we usually radiograph the portions of their body parts in a standing condition, and wherein the cassette hole a little far off distance and expose the body part. So while doing so, the personnel also have to around 0.5 mm lead, which will protect the eyes of the personnel while holding or this is a screen that you see. This is also having the lead impregnated wall or a shield, impregnated wall or a sheet. This is a shield through which you can see there is a window. So while shooting an X-ray, the personnel has to look at this. This glass is also lead made of lead, and while shooting an X-ray, the personnel has to look through this lead glass and shoot so that the minimum exposure to the personnel would happen. The other things that the personnel while shooting an X-ray has to wear, it is the lead gloves. It is very, very important to protect the hands or the arms, and it comes in at various different colors of a different shape and sizes. One can choose different colors. The colors are based on the numbers, that 704, right from 710, 712, 705, 713, 718, 719, and so on. The goggles and the aprons, basically, this has to be worn by the personnels who are working in the radiological section. They must wear this apron, and there will be a thyroid shield that has to be worn around the neck to protect the thyroids. Those are radio-sensitive organs, and these lead devices has to be hung on the hangers, because if you just fold them, the lead impregnated in between will get broken or will breach it, and the very purpose of protecting, receiving a protection from the X-ray will get hampered. The last thing that is very, very important to understand is the thermoluminescent dosimeter, which is a device which is being given by the BARC, Bhabha Atomic Research Center, to all the organizations having an X-ray devices, and to all the personnels who are working under in the area of radiological section, needs to be distributed to them, so that they can wear it around their neck, they can wear it around their wrist, they can wear it around the waist. Okay, and the routine regular monitoring would happen once they regularly work in the area for around 4 to 6 months. These batches then are taken from those personnels, given to the BARC for giving. Upon receiving, they will calculate the dose, or they will declare the exposure factors or exposure, radiation exposure for those personnels, and based on that they can always declare the danger line, that this kind of a person who is having a particular exposures doses must be changed from that point of posting to the another point of posting, so that a kind of a rotation shall always happen, and it shall be taken into consideration, so that the harmful effects can be prevented, especially for those who are working in those areas having radiological devices as being used as a part of the diagnosis or as a part of therapeutics. So with this, I think this is enough to talk about the radiation sensitivity of different body parts and the radiation production devices. With this note, I thank all of you for your patient hearing. Thank you so much.